Hi, this is Dr. Paul Sachs, and welcome to our Q3 quarterly brief on HIV medicine. Joining me today is Dr. Raphael Landowitz. He is professor of medicine at UCLA, and he is also a longtime friend. So, Rafi, welcome.
Thanks, Paul. Pleasure to be here.
So, what we're going to do is start by focusing on a bunch of studies that were presented at the IAS conference this past summer. And I want to say that it was kind of held in Berlin, although a lot of stuff is virtual these days because of COVID, but there was an actual place in Berlin where they held this conference. And it reminded me of the Berlin conference in 1993, which is actually the very first conference I went to. That was the 9th International AIDS Conference. And the difference is really staggering. That was first one of the most depressing HIV conferences ever. And I was able to find actually a Los Angeles Times review of that conference saying that it was so discouraging because it was found in the Concord study that the AZT that we were using probably didn't work in people who didn't have symptoms. And also there weren't a lot of promising drugs in development quite yet. And so comparing that to what we have now with really unbelievable options for therapy really showed me how much incredible progress we've made in this disease. We're going to start off now by talking about HIV prevention. And it's really an honor to have you here, Rafi, because you're one of the top experts in this area. So why don't you take us through our first studies?
Thanks, Paul. The first thing I'm going to cover is some updates on HPTN 083. I know on previous versions of this webcast, you've covered the early results of this study, which was a randomized phase 3 study comparing long-acting injectable cabotegravir administered as a gluteal injection every 2 months to daily oral TDF-FTC for pre-exposure prophylaxis in cisgender men and transgender women who have sex with men. And really, what's new in the last couple of months is we were able to finally publish the primary results of that study in the New England Journal. And just to remind people, what we saw was that although cabotegravir was superior to TDF-FTC in that trial, my interpretation of those results is really what we saw was an effectiveness result. And what's clear from 083 is it covered sex acts better in high risk, particularly young transgender minority populations. So that's my interpretation of the results. We're still trying to figure out onset of protection, offset of protection. Do you need an oral lead-in? And importantly, we're trying to figure out this observation that these long-acting potent antiviral agents in the prevention context seem to obfuscate conventional HIV testing algorithms. They delay the reactivity of these conventional algorithms we use. So there's some new challenges to diagnostics that we're going to need to figure out. So lots to be clarified as we continue this study. It's now unblinded, and we are making the oral lead-in optional for people who are now coming into cabotegravir for the first time, and we're implementing sensitive RNA-based testing as part of the screening algorithm to get some data on what the utility of these more sensitive viral load or NAT-based tests might be. Before I ask you, Paul, what you sort of think about all this and what you think it means to the field, 084, our sister study, which looked at a very similar study design in cisgender women in sub-Saharan Africa, also had a stunningly impressive result. They were stopped by their DSMB about 6 months after we were. And so particularly with the challenges of finding cisgender women who are interested, able, and willing to take an oral preparation and getting population level efficacy there for prevention, this is really, really exciting and could be transformative for cisgender women, increasing options. But Paul, what are your concerns about this? Because you do this in the clinical setting all the time, and this testing thing is really a bugaboo, isn't it?
Well, I have two thoughts and one question for you. One is that the testing part is going to keep us in business for some time because these are incredibly complicated cases. These possible breakthroughs, and I always go back to Jean-Michel Molina's review of ambiguous test results in the context of PrEP. That review he did at CROI a few years ago, and then also a really nice paper that the CDC group put out. But it's really not clear what to do when you have someone on PrEP and they suddenly have a positive test and you're not sure whether it's a true positive or false positive. Very difficult. So that's one thought. The second thought I have is we now have these two large randomized clinical trials where one strategy is superior to the other. And yet, as you noted, it would not have been superior if you only include people who were adherent to TDF-FTC. And so that raises the question of, and this is the question back to you, person comes in, generic person, at risk for acquiring HIV, wants to be on PrEP. What are you telling him or her is the best treatment to go on?
What would I recommend to somebody who's coming in? Two things. I think if somebody is able to take TDF, FTC, PrEP, either pericoidally, if they're an MSM, or daily, no matter what the population or risk is, to me, that's a tried and true proven intervention. And unless there's some compelling reason to do something different, I think that's what I would recommend. If there's any question either in a provider or a consumer's mind about their ability to adhere to that regimen, I think cabotegravir has demonstrated that it works, and it works really well. We just don't have as much information about why it doesn't work when it doesn't work.
Yeah. Okay. Thank you. Thank you for being concise. I know you could go on for a while about that, and appropriately so. But it raises an interesting dilemma, because normally when we have something that's superior to something else, we immediately shift the standard of care, but that's not going to be so easy. I also think there are significant pharmacoeconomic implications, because TDF-FTC, of course, is generic. All right. What other studies do you want to cover? Prevention.
Thanks, Paul. Just two other things that I thought were important to highlight. There was the CRO-PrEP study, which was a study in 4 Chinese cities that looked at the effectiveness and adherence of tenofovir-based pre-exposure prophylaxis in MSM in China, and they looked at the incidence rates for HIV in people who were taking a tenofovir-based PrEP orally or on demand. And what they found were extremely low incidence rates in both arms. These were people who chose. This wasn't randomized. And what was really interesting and nice was they also followed a similarly sized cohort of PrEP refuser MSM as well, to get a perspective for what the incidence rate was in the background population, which isn't something that we often have access to. And that incidence rate in this population was about 5.1 per 100 person years, which is, it certainly makes you sit up and take notice.
Yeah, I thought that was a great aspect of the study, that they were able to do that.
Interestingly, the absolute incidence rate in the on-demand arm was actually lower than in the daily arm, and they noticed a trend towards decreased appropriate coverage of sex acts in the people who chose the daily option compared to the on-demand option, which I think raises an important issue, that you want interventions that are going to be sexually congruent with people's behaviors. And so, having people understand the ability to swap between those two ways of taking tenofovir-based PrEP is probably important as we educate people about its use going forward. And then the last thing I just want to really briefly just touch on, because I think there's a lot of misconceptions about what these data mean or how ready for prime time these data are. Jenna Yeager from Pete Anderson's group at University of Colorado put out a really elegant paper where they did DOT dosing of TAF-FTC and TDF-FTC, either in-person observed dosing or video observed dosing. And they did some really elegant pharmacokinetic studies, and they looked at PBMC tenofovir diphosphate levels of 3 different dosing schedules: people who took each product every day, people who took it one day and then not the next 2 days and repeated that, and then people who took it day 1 and day 2 but not day 3 and repeated it. So really, 1/3, 2/3, 3/3 kind of dosing comparison. And what they found, and this is really interesting, is the intracellular tenofovir diphosphate concentrations for TAF FTC taken 1 every 3 days were actually higher than the 3 of 3 days for the TDF FTC. And so some people have extrapolated that to say, really, two doses a week of TAF FTC is more effective than daily TDF FTC. And, maybe that's going to turn out to be true, but we're not there yet. And remember, one of the biggest biblical or Talmudic discussions in pre-exposure prophylaxis or biomedical prevention is, what's the right pharmacokinetic correlate of prevention? Is it intracellular levels? Is it plasma levels? Is it tissue levels at the site of exposure? And on top of that, how do sort of ulcerative diseases at the mucosal location of exposure going to impact that efficacy and those concentrations, that pharmacodynamics, really. So just, people are going around saying this is a done deal. It's not a done deal. This is fascinating. It likely suggests that there are important differences between the two agents. What they exactly mean yet, it's premature to say. So that's sort of my wrap up of the prevention. I don't know if you have thoughts on either of those two other studies,
well,
Paul.
One of the most common asked questions in practice is, can you use TAF FTC for on-demand prep?
Yeah, we don't know yet.
Okay. All right. Well, that's thank you for that incredible summary. And we'll see. We know injectable cabotegravir is around the corner for PrEP, and it's not FDA approved yet, but by the end of this year, it should be. Now on to treatment. I wanted to start by talking about a study called the STAT study, and it actually was presented by Charlotte Page-Roll. Basically, what it looked at is a single-arm study of just over 100 people newly diagnosed with HIV. Can you give dolutegravir lamivudine as initial therapy? That's the question they asked. And this single-arm pilot trial found that indeed this was highly effective. Only 9 of the people in the study had to change based on having transmitted M184V or hepatitis B. And it depends. It's a glass half empty, half full kind of result. I'll give you the glass half full first, okay? The glass half full found that if you take a look at the as-treated population and eliminate missing data and loss to follow-up and et cetera, then it was a 97% successful strategy. That's glass half full. The other glass half full is it didn't seem to matter how high the viral load was. People with viral loads of over 500,000 did great. People with viral loads over 1 million did great. So that's the glass half full. The glass half empty is that there were a bunch of people who didn't complete the study. And so the average, the efficacy out at week 48 was like 80%. And then the other part is that they gained a lot of weight. The weight gain was actually substantial, and even a lot compared to some of the studies that we've seen with TAF FTC dolutegravir. So clearly, TAF alone is not driving the weight gain. And then the final thing is, and this is the biggest question for dolutegravir lamivudine as an initial strategy for rapid start, is what's gained by doing this? And that is, I'd say, the biggest question of all. And do you have any thoughts?
I do think that's the $1 million question, right? What do you lose by using a 3-drug regimen right away? We have highly effective single pill, well-tolerated regimens, and you can always back off once you get that initial genotype and the hepatitis B testing back. Because the other thing that I'm sure you noted was, 7 of these participants actually had chronic hepatitis B, and they only had resistance testing available after treatment, after that finding was unearthed in 3 of them, and they didn't get hep B resistance with that short-term treatment. But 4 of them, we don't have any information. And it seems to me like, what does this get you?
Yeah. No, I think that's the biggest question, because I believe it virologically. I do. I think this is an extremely effective regimen, but it doesn't seem to fit this particular need of this person who starts the day you see them. Going to move on to another study, and this is one of those, quote, long-term follow-up studies, and I was involved in some of the original studies with bictegravir FTC TAF. And again, we're combining 2 studies. One is the study that compared it to to TAF-FTC dolutegravir, and the other study it's comparing to abacavir 3-TC dolutegravir. And, we're out now at something like, what is it, 192 weeks. So that's, just in case you're doing the math in your head, it's 4 years. That's right. Were you a math major? No.
Chemistry.
Chemistry. Okay. I knew it was some science. Anyway, it's a lot of weeks. And the amazing thing about our current regimens, and this goes back to what I said about the Berlin 93 conference, is that, again, as treated, 99% of the people in the study have viral loads less than 50, which is just amazing. Now, of course, if you take the missing people out, it's down to 75%, but that's typical for 4 years of follow-up. Not everyone's going to stay living in the same town, but 99%, not only that, but no resistance. It's really remarkable. I should say that the comparative parts of these studies ended long ago. Now everybody in these studies is on bictegravir FTC TAF. And these results kind of explain why, these days at least, bictegravir FTC TAF is the most commonly selected initial regimen. So it's just it's good to have long-term follow-up. And yes, weight gain does occur in people starting therapy with this regimen. So let me move on for the sake of time and now go back to dolutegravir lamivudine, because I want to talk about switch studies. The SALSA study was presented for the first time. And the difference between the SALSA study and the TANGO study, because they both included virologically suppressed people who had never failed treatment before, is unlike in the TANGO study, where everyone was on a TAF-based regimen at baseline. In the SALSA study, they actually were on a variety of different regimens at baseline. It's a standard triple therapy, but they could be on, a integrase inhibitor, a non-nuke, or a PI plus TDF-FTC or TAF-3TC or whatever, or TDF-3TC or TAF, you named it. And I'd say about 50%, just under 50% of people were receiving TDF at baseline. And I think that's important when we look at the results. First, the virology results. The virology results are just, outstanding. People who are suppressed and have no resistance can switch to dolutegravir lamivudine with really essentially zero risk of virologic failure. And I think we should stop being surprised about this. And there was nobody who actually ended up getting resistance, because there were so few virologic failures to begin with. So that's the virology part. It's boring in a good way. The next part are drug-related adverse events. And here, they were actually higher in the dolutegravir lamivudine arm than in the people who continued therapy. And this is one of the hazards of doing an open label trial, because people who are stable on their current treatment and switch to something new, if they get any sort of side effect, they're going to blame it on their new treatment. It's the so-called ascertainment bias. And we've seen it time and time again. And so if I were to redo the study, I would do it blinded, but then it's so hard because the baseline regimens were so different. But nonetheless, that's what they did. So that means that there were more adverse events in the people who switched. And the adverse event that was actually quite notable was weight gain. And why do you think that happened? People, were not necessarily worried about this, and yet they go into this study, the SALSA study, and they end up reporting weight gain as a side effect leading to regimen change in 6% of the switchers versus none of the people who stayed on their regimen. And I think what we're seeing is an accumulation of evidence that anything that leads to a stopping of TDF is going to lead to weight gain. And TDF as this weight suppressant, who knew? That is emerging as a dominant risk factor for weight gain when people switch therapy, especially if they're receiving efavirenz, and several of the people in this study were. So anyway, virologically great, but when you do this switch, especially if you're switching people off TDF, just be concerned about that particular side effect.
Well, remember, we saw that too in the HIV uninfected PrEP studies as well.
Absolutely. And, in fact, one of the really, it's really interesting to observe this in hindsight, and people aren't really sure of the mechanism. Is it, quote, an adverse event, meaning it was causing some sort of nausea, or is it actually doing something beneficial? I have no idea. I can't tell you the answer to that. So that takes us now to the DOLAM study, which was actually just published in the Lancet HIV. And here is another non-inferiority switch study that is totally acceptable for people to switch to dolutegravir lamivudine, no excess virologic failures, no excess anything. And it really is just sort of confirms what we've learned from Tango and Salsa, that this is a safe study to do from a virologic perspective. I want to actually now shift away from the dolutegravir lamivudine switch studies and talk about a very important study that was just published in the New England Journal of Medicine called NADIA. This is a study that looked at people who failed first-line treatment in Africa. And first-line treatment is a double nucleoside plus a non-nuke, and the double nucleosides tenofovir 3TC. And what's different about Africa versus the US is that when they fail treatment, they don't necessarily switch right away. So they can have prolonged periods of viral replication, selecting for more and more resistance. And the astounding thing about the NADIA study is the amount of resistance they found, in particular in the nucleoside analog class, so that a substantial fraction of the people in the NADIA study, when they entered into it, already had complete resistance to the nucleosides. So they had M184V and they had K65R, or they had, who knows what, lots of other mutations. And then they were randomized to 4 different strategies in a what's called a factorial design. One strategy would keep the tenofovir 3TC, just keep it. And the other would switch it to zidovudine, because remember, that's the WHO recommendation, and zidovudine is active against K65R. And the other randomization was to either dolutegravir or to darunavir ritonavir. So everybody in the study gets at least one new drug, and then some just repeat the old nucleosides, and some get 2 new drugs because zidovudine is a new drug. And it turns out that they were all equally effective, which is remarkable. And so even when you looked at the subset analysis of the people with 0 active nucleosides, the tenofovir 3TC arm did as well as the zidovudine 3TC arm. So lessons learned. 1, dolutegravir has an incredible resistance barrier. It's as high as a boosted protease inhibitor. I don't think we need to make apologies for it anymore. 2 is that the nucleosides are definitely still active, even when our resistance algorithms say that they're not. And 3, big question, it relates to bictegravir. Can you do the same thing with bictegravir FDC-TAF? Because that's obviously a simpler treatment and one that doesn't have any TDF, renal toxicity, bone toxicity. So your thoughts about that last one?
My guess is yes. My guess is you can, but it would be great to have data to support that. But the other question, Paul, that you didn't address that I sort of just gives me a little bit of anxiety is, right, we know from a number of studies that dolutegravir monotherapy looks good up front, but the longer you follow people, the less fabulous it looks. We have fairly limited NADIA follow-up at this point. Do you think that we're going to see that equivalence between darunavir and dolutegravir stay parallel or diverge a little bit the longer we follow?
Actually, I maybe overstated the resistance barrier issue with dolutegravir, because there were some patients in the NADIA study who developed dolutegravir resistance. I think there were 4. And as a result, perhaps the real Achilles heel of this strategy will be that subset of people who are not quite adherent enough to the dolutegravir regimen to maintain suppression, and they get dolutegravir resistance. But it's not going to be as bad as dolutegravir monotherapy. Let me move along. I wanted to talk about the capsid inhibitor studies, and it's fascinating. A new drug class is coming out, first of its kind. And we have a phase 2 study in treatment-naive patients, and then they get assigned to one of 4 regimens. One of the control regimen is easy; it's bictegravir FDC-TAF. But then the capsid regimens, which the capsid inhibitor is called lenacapavir, has actually lenacapavir given subcutaneously every 6 months. That's a lot. That's infrequent. Or in 2 of the arms, and then the 3rd arm gets lenacapavir orally, and then that's accompanied by TAF FDC. And then the plan eventually in this study is to give lenacapavir with just one other drug, either tenofovir alafenamide or bictegravir. So you have your 4 arms there. 2 of them have a subcutaneous lenacapavir every 6 months. And one of them has lenacapavir orally, and one of them has bictegravir FTC TAF. And as a phase 2 study goes, the patient characteristics, they weren't that advanced because you don't want to have people with advanced HIV disease in your phase 2 study. And the results overall showed that just under 95% of the people in the lenacapavir arms were suppressed at week 28, and 100% of the people in the bictegravir arm was suppressed. And that sounds good, but there's this one niggling issue, and that there's one patient in the treatment group 2 who got the Q6 month who developed resistance to lenacapavir and to the M184V, so emtricitabine. And I just got to say, it's interesting to see this. The patient had baseline viral load of about 100,000. They measured the lenacapavir levels. They were in the expected range. They didn't present data on dried blood spots of tenofovir concentrations. I don't know what the adherence was, but the patient has subsequently been virologically suppressed on a dolutegravir regimen. So it's just one patient, but it's still, I don't know. What are your thoughts?
Well, I think, you're going to, give us an update on Capella also, but similarly, there's this whiff, this little hint that maybe lenacapavir doesn't have a particularly high barrier to resistance. And is that going to be an issue? Are we going to need second generation or third generation capsid inhibitors before this is able to realize its potential? Certainly the administration interval of every 6 months is extraordinarily attractive. But is it going to have the antiviral durability that we're used to seeing with these daily dosed agents? I think it remains to be seen. That one case similarly gave me pause also.
Yeah, I mean, it's something that we didn't see in any of the dolutegravir or bictegravir studies, as I mentioned. And then you mentioned Capella. We did see these data before. It's a very treatment experienced patient population. Whenever I talk about treatment experienced patient populations, the first thing I go to is what proportion of the patients in the study had integrase resistance, right? And it's actually 70% had integrase resistance. So we're talking very significantly treatment experienced. And with that background rate of resistance, small numbers, but they were able to randomize to a lenacapavir arm or, and then there's a non-randomized group who didn't really have any active agents in background. And the study design is somewhat complicated, but even with these small numbers, I'm going to mention that the overall results definitely imply an active drug that can actually lead to viral suppression in a substantial fraction of them. 29 out of the 36 or so had viral loads less than 50, and then a higher proportion had viral loads less than 200. So if there's any doubt at all about lenacapavir being an active agent for this population, I think that should be, don't worry about it. So this agent is, because of this study and because of the need for active drugs for patients with no options, this has actually already been filed for this population. And as you alluded to, Rafi, there were some patients who developed lenacapavir resistance, and that was in the randomized cohort. We saw, I believe it was 4 patients, 4 out of the 36, who developed lenacapavir resistance, and then they've been subsequently suppressed in management. So lenacapavir as a new drug for this small subset of patients with no options, I believe it'll be a nice thing to have, especially because ibalizumab is a tricky drug to give. We have fostemsavir; it might be able to be combined with that. Doesn't come up that often, but it will be useful. Now, why don't you just wrap us up with a couple more comments about these drugs in PrEP, and then we'll finish.
Thanks, Paul. At IAS this summer, we did get to see some macaque challenge data, this time vaginal challenge data for sort of the analog drug of lenacapavir called GSCA1, which is a slightly different capsid inhibitor that Gilead developed and is used in their non-human primate models. So just to be clear, this isn't lenacapavir. It's similar to lenacapavir, but it's not the same product. And we had previously at CROI this year seen rectal challenge data in a similar model, single subcutaneous dose of this capsid inhibitor, and then serial rectal, this now vaginal challenges, high levels of protection against HIV acquisition with the highest dose, which was 300 milligrams per kilogram. Also potent efficacy at half that dose, but better efficacy at the full dose. So again, supporting the ongoing phase 3 clinical trials that are evaluating lenacapavir as PrEP across a variety of populations that are currently in the field and are eagerly being anticipated. And then speaking of agents for PrEP that are in the field in advanced stages of development, Sharon Hillier gave us the unblinded safety data from the 016 study, which is a phase 2A, so low risk participant trial of monthly islatravir at either 60 or 120 milligrams. We had previously at R4P this year seen the blinded aggregate safety data and PK, and now they've unblinded, so we have a little bit more of a sense of the side effects of this drug, and it was generally well tolerated. I think this is honestly the long-acting drug that I'm most excited about seeing the results for. It's in advanced stage phase 3 trials across a variety of populations globally, and we're looking forward to results. I think, a monthly oral pill that could be given directly observed therapy or, on the first of every month kind of thing is potentially very powerful.
So, Rafi, I want to thank you for this great conversation for our Q3 quarterly brief. I hope that you will join us again sometime in the future. Take care.
Thanks so much for having me.
Thanks to all of you for listening.